Hey guys, Miss Cote here, and this is episode number 5 of the cell unit. This is the second video in the cell transport chapter. In the last episode, we looked at the structure of the cell membrane in detail, focusing on the function of the main components: phospholipids, cholesterol, proteins, and glycoproteins. This episode will start focusing on how materials can move across the membrane, more specifically through passive transport. Remember, the way the cell membrane is set up, it's very selective about which molecules can cross without help and which molecules require some assistance from membrane proteins. We call this being selectively permeable. So let's get started. Let's look at this model for a minute to reacquaint ourselves with the structure of the cell membrane. Remember that the cell membrane has two regions to it: the hydrophilic heads, which like water, and the hydrophobic tails, which don't like water. The biggest obstacle for molecules would be the span of hydrophobic tails in the middle part of the membrane. So if you're a non-polar hydrophobic molecule, it will be very easy for you to cross the cell membrane, and you wouldn't require any assistance. However, if you are polar, or a molecule with a charge, otherwise known as an ion, then you would have hydrophilic properties. This will make it hard for you to cross the membrane and would need the assistance of a transport protein. Transport proteins allow the passage of hydrophilic molecules across cell membrane. These proteins can be of two types. Channel proteins on the left create a tunnel for the hydrophilic molecules to pass through. It basically shields the molecules from the hydrophobic part of the membrane. The other type of transport protein is called a carrier protein. When a molecule binds to a site on the carrier protein, it changes shape and shuttles the molecule to the other side of the membrane. Just know that these proteins are specific for the type of molecule they transport. Let's start looking at passive transport. Remember that molecules are constantly in motion, and this motion is completely random. When these molecules bump into each other, they'll change direction. So if there are a lot of molecules in a given area, there will be a lot of collisions and changing directions. Every once in a while, a molecule moves in a direction away from the other molecules. This is diffusion happening. Diffusion is when molecules spread out evenly into an available space. And once equilibrium occurs, the molecules still move, but they move evenly on both sides of a membrane. Here on the top, we have an example of diffusion of one substance: molecules of dye, like food coloring. Random movement of the molecules will cause colliding and changing directions. Every once in a while, a molecule will pass through a pore in the membrane, sending it to the other side. Once equilibrium is reached, for every molecule that moves to the left, another moves to the right. The bottom shows an example of diffusion of two solutes. While the yellow molecules randomly diffuse over to the right, the purple molecules will do the same, but to the left, until equilibrium is reached. And still, even at equilibrium, the molecules are still moving, but there is no net movement of either molecule on either side of the membrane. We just saw that molecules will randomly move from an area where they are more crowded, otherwise known as a high concentration, to where they are less crowded, known as an area of low concentration. The difference between the two areas is referred to as the concentration gradient. In this case, going from high to low concentrations is described as moving down the concentration gradient. And because this is due to random movement, it does not require any work. It just happens naturally, so no energy is needed for this to happen. That is why we call it passive transport. Osmosis is another example of diffusion, but it is specifically for the diffusion of water molecules across a selectively permeable membrane. Water is going to move from an area of high concentration to an area of low concentration, or like the slide says, from an area of low solute concentration to an area of higher solute concentration. But if you find that confusing, just focus on the water molecules. So just like diffusion, no energy is required for osmosis to happen. This contraption here is called a U-tube. not the channel you're watching this on, the actual letter U, like the shape of the tube. Here we have two different solutions separated by a semi-permeable membrane. The solute, represented by the green dots, is too large and cannot pass through the pores in the membrane, but the water molecules are small enough to go through. So in this particular instance, it's the diffusion of water that's happening, not the diffusion of the substance. So we're discussing osmosis in this case. Looking at the U tube, you can see that the solution on the right is more concentrated, it has more green dots, than the solution on the left. But the one thing that these pictures usually never show you is the water molecules themselves. You just have to be able to visualize them. If we look at a close-up of what's happening around the membrane, the water molecules are now represented. You can see that on the left, where there is less of the green solute, there are more free water molecules than on the right. I always think about it as the less stuff, the more water, and vice versa. Anyway, if we allow this apparatus to sit, what will eventually happen is that the free water molecules will start moving randomly from the left side to the right, from where they're in a higher concentration to where they are in a lower concentration. Or you can say that they are moving down their concentration gradient, and this will cause the water level to rise on the right side of the U tube. There are some vocabulary words that you should be familiar with when talking about water balance. A cell can either gain water or lose water, depending on the type of solution it's surrounded by. This is called tonicity. You'll need to understand how to use these terms when you're comparing two environments of differing concentrations. You can pause this slide to write down the terms and definitions, but I'm going to explain these terms with the figures on the next slide. Here we have an example of both an animal cell and a plant cell under the three different conditions. Let's start with the cells without a cell wall. An isotonic solution is one that has the same solute concentration as the cell. Since the cell and its surroundings are in equilibrium, there is no net movement of water across the membrane. For every water molecule that goes into the cell, there's a water molecule that leaves. As far as the cells are concerned, they basically stay the same, which is good for our cells. That's the environment they want to be in. If the cell is placed in a hypertonic solution, that means there is more solute in the solution than in the cell. Hyper means more. As a result, the water will move out of the cell into the solution, moving from high concentration to low concentration, and the cell will shrivel up and most likely die. This will become an issue if you're a freshwater organism that is placed in salt water. Or, I don't know how many of you have seen that cruel video of what happens when you put salt on a slug. If the cell is in a hypotonic environment, then the solute concentration in the solution is less than that of the cell. So if the water moves from high concentration to low concentration, then the water will be moving into the cell. If the cell does not have a cell wall, then it will pop or lyse. This is an issue for organisms living in freshwater environments. Water will constantly be moving into the organism. Because of this, organisms have developed certain adaptations to maintain water balance. For example, Paramecia have structures called a contractile vacuole that will continually squeeze out the water that enters the organism. Maintaining the water balance in organisms is called osmoregulation. If you would like to learn more about some adaptations that organisms have for dealing with water balance, you can read about it in chapter 44. Okay, now we should talk about the water balance of cells with cell walls. The optimal environment for a plant cell would be in a hypotonic solution. While this would cause an animal cell to burst, the cell wall of a plant cell keeps it from lysing. Instead, the cell being full of water puts pressure on the cell wall. We call this turgor pressure, and the cell is said to be turgid. This is the healthy state for plant cells. In an isotonic environment, when there is no net movement of water, the turgor pressure is lost, and the cell is referred to as being flaccid. In a hypertonic environment, just like an animal cell, the water is being lost from the cell and the cell membrane pulls away from the cell wall. This is called plasmolysis. At this point, the whole plant is wilted and will most likely die. Here's the slide with the vocabulary for water balance in plant cells. You can pause the slide to copy down the vocabulary if you need to. We've discussed diffusion and osmosis. The last example of passive transport is called facilitated diffusion. This type of transport is for those molecules, the polar and charged molecules, that need the assistance of transport proteins. Let's go through the two types of transport proteins. Channel proteins provide a tunnel for a specific molecule or ion to get from one side of the membrane to the other. For example, sodium ions have their own channel protein, and chloride ions have their own separate channel proteins, specific proteins for specific molecules. These proteins are called ion channels, quite simply because they move ions across a membrane. Some of these ion channels are always open. However, some are referred to as gated channels. They open or close under certain conditions. There's another type of channel protein called aquaporins. These are channel proteins that are specifically for water. While water is small enough to move through the membrane by itself, aquaporins allow the water to move faster and more efficiently across the membrane. So while water can move across a membrane through osmosis, it can also travel through facilitated diffusion. The other type of transport protein are referred to as carrier proteins. These proteins change their shape in response to binding of the molecule and moves that molecule across the membrane. An example of a molecule that uses this type of protein is glucose. Glucose, being a polar molecule, needs to be shielded from the hydrophobic part of the membrane, and the carrier protein accomplishes this. It moves glucose from a high concentration to a low concentration, and even though the protein changes shape, it does not require any energy in order to do this. And that's it for passive transport. We'll continue this discussion of cell transport by looking at active transport in the next episode. If you have any questions about passive transport, make sure to write them down and bring them up during class. So until next time, be a good human.